Hello everyone, I'm Komal. Welcome to InGrade learning platform JEE NEET Foundation, where you learn, practice, crack, succeed. Stay focused, stay unstoppable. Okay students, so last time हम लोगों ने न्यूक्लियोटाइड्स फिनिश कर लिए थे, न्यूक्लिक एसिड्स हमारा फिनिश हो गया. Now we have to start with enzymes. So today we'll start with the enzymes. Okay. So what are enzymes? What are enzymes? As I've told you in the structure of proteins, when we did the level of organizations in proteins, I've told you there are 4 types of organization. One is primary structure, secondary structure, tertiary structure and quaternary structure. And enzymes are considered under tertiary structure. Okay, so what are enzymes? They are basically proteinaceous substances. They are proteinaceous substances that are capable of catalyzing biological reactions. That is why they are also known as biocatalysts, hence known as biocatalysts. Okay, see, like our environment, we know in our environment there are many of the chemical reactions occurring. Similarly, in our biological bodies, whether in plants, animals, bacteria, viruses, there are millions of biochemical reactions, there are millions of biological reactions that are occurring in our body. Okay, so these chemical reactions may take a lot of time if there is no enzyme present in our body. Okay, so to make their rate of reaction faster, we have enzymes. Okay, so that's why they are known as biocatalysts, because enzymes are basically catalyst only. Okay, okay. Next. Okay, enzymes are mostly proteinaceous. Enzymes are mostly proteinaceous. But we have few exceptions. What are the exceptions here? We have 2 enzymes which are not proteinaceous, instead they are RNA enzymes. Okay, so the examples are ribozyme and second is ribonuclease P. They both are RNA enzymes. Okay class, next. Now let's study about the difference between enzymes. Let's discuss about difference between enzymes and inorganic catalysts. Let's see. If we talk about enzymes, okay, and inorganic catalysts which occur in the nature, occur in our environment. Okay, so enzymes are basically, if we talk about their structures, right? If we talk about their structures, then we see that enzymes are complex molecules. They are complex macromolecules. And if we see about inorganic catalysts, we see that they are simple molecules or mineral ions? If we say about the temperature, if we talk about the temperature, then enzymes cannot withstand high temperature. Cannot withstand temperature more than 40 degrees Celsius. Whereas inorganic catalyst can withstand higher temperature. Can withstand high temperature. Okay, if they cannot withstand, the enzymes cannot withstand more than 40 degrees Celsius. If the temperature goes above 40 degrees Celsius, they get denatured. Okay, enzymes are highly efficient and they are less efficient. Okay, if we talk about the specificity, enzymes catalyze only specific reactions. है ना? Every chemical reaction, every biological reaction have specific enzyme to catalyze them. Okay, Rizwan, hi beta. Ma'am, do you have offline classes? Beta, please contact this number to get more details. Ma'am, how many hours should we study each day? Yes, good question, Rizwan. To crack the NEET exam, I think you must study. You must give at least 3 hours to every subject per day. So basically, you need to study at around 8 to 9 hours per day. Okay, Rizwan, I hope you have got your answer. Please comment down. Okay. Next we have is, okay, so basically enzyme are highly specific. Specific enzyme catalyze specific reaction. Okay. Whereas if we talk about inorganic catalyst, single particular catalyst can catalyze numerous reactions. One more difference we are having here is they occur in living organism and they do not occur in living organism. Okay, class. Okay, one more thing. If I talk about chemical reactions, no, not chemical reactions. If I talk about the temperature, okay, so we have enzymes. Generally, enzymes cannot withstand. Generally, enzymes cannot withstand high temperatures wait. Let me create a space. Have space here. Okay. So see, most of the enzymes cannot withstand temperature more than 40 degrees Celsius. More than 40 degrees Celsius. Otherwise, they get denatured. Otherwise, they get denatured. But we have some enzymes which are isolated from thermophilic organism. What are thermophilic organism? Which live under high temperature conditions. For example, we have some bacteria which live in sulfur springs, hot vents, है ना? So they must have enzymes which can withstand high temperatures. So we can isolate those enzymes, and those enzymes have specific characteristic, that is thermal stability. So we can use them. Okay. But some enzymes, but enzymes isolated from, but enzymes isolated from thermophilic organisms which live under high temperature, जैसे कि सल्फर, hot vents, have thermal stability. Okay, and one such enzyme we can get is tech polymerase. Example of this enzyme is tech polymerase. Let me write some. Example of such enzyme is tech polymerase. Okay, isolated from Thermophilus aquaticus. Okay, class. Okay, now let's study about the chemical reactions. Let's study about the chemical reactions. So we have chemicals go under two types of chemical reactions. Chemicals or substances undergo two types of chemical reactions. One is physical reaction. Second is your chemical reaction. What is physical reaction? What is physical reaction? That do not basically type of physical reactions are like change in shape or state. Change in shape or state. So they do not require making or breaking of chemical bonds. Okay, class. And if we talk about chemical reactions, okay, it involves involves making and breaking of chemical bonds. Okay, in this also we have two type of chemical reactions. One is organic, second is inorganic. We have two types of reaction. One is organic, second is inorganic reaction. If we talk about organic reaction, we have example of such as, if we talk about starch, when we hydrolyze them, it becomes, we have to use the enzyme here amylase, and it becomes glucose. Okay, after hydrolyzing it. And if we talk about inorganic reaction, your NCERT tells us one reaction, that is barium sulfate. Barium sulfate, sorry, not barium sulfate, barium hydroxide, barium hydroxide plus sulfuric acid forms barium sulfate, and it releases two molecules of water. Okay. Now, if we talk about rate of reaction, what is rate of reaction? What is rate of reaction? Amount of product formed per unit time, that is delta P upon delta T. This is known as rate of reaction. Basically, rate of reaction is the speed of reaction. Okay. So, if speed has definite specified direction, if the rate of reaction have specified direction, okay, so it is also known as velocity. It's also known as velocity. Okay. Okay. Now, mainly the rate of reactions are influenced by rate of reactions are influenced by temperature, and we have a general thumb rule. What is the thumb rule? That if temperature increases by 10 degrees Celsius, rate of reaction doubles. And if temperature decreases by 10 degrees Celsius, the rate of reaction get half. Okay, class. Now, presence or absence of enzyme makes a lot of difference. Okay. For example, if we have a simple reaction, say a simple reaction, that is CO2 plus H2O forms H2CO3, that is carbonic acid, which requires carbonic anhydrase enzyme. it tells you now condition, NCERT tells you the condition that if there is absence of carbonic anhydrase, how many molecules will form, and if there is presence of carbonic anhydrase, how many molecules. Okay, so let's see. If we have absence of enzyme, or carbonic anhydrase, only 200 molecules per second get formed. Okay, 200 molecules of H2CO3 get formed per second. And if we talk about if we have enzyme present, okay, then we have 6 lakh molecules formed. Not second will come, per hour. And if we talk about in presence of enzyme, we have 6 lakh molecules get formed of H2O3 per second. Okay, so basically rate of reaction got increased by 10 million times. That's why enzymes are very important to have in our biological bodies. Okay, class? Okay, now what are metabolic pathways? What are metabolic pathways? What are metabolic pathways? See, we have thousands of enzymes. We have thousands of enzymes catalyzing a specific reaction. Catalyzing a specific reaction. Okay. So, when multi-step reaction occurs, multi-step reactions are catalyzed by same or different enzymes, Okay, for example, we have, if we talk about the glucose get converted into 2-pyruvate. Now this process, the conversion of glucose to pyruvate, need 10-step reactions. This is basically what it is, a metabolic pathway. Okay, class? Okay. Next we have is Activation of energy. How enzymes work basically. Let's see how enzymes work. Let's see how enzymes work. Students, we have basically what we have is see, we have enzymes. Enzymes are basically tertiary structures. Okay, they are tertiary proteins. They are tertiary proteins. Okay, so the backbone of protein, backbone of protein gets folded, and forms pocket-like structures, pockets called active sites. Okay, now to these active sites, some substances known as substrate gets attached. Okay, suppose we have this substrate. Enzymes have active sites which substrate gets attached, and they form transient states known as enzyme substrate complex. The transition state. These are transition states. Okay, these are transition states, and when substrate gets bounded with the enzyme, the making and breaking of bonds occur, and substrate gets converted into products, and the enzyme gets released unchanged. Okay, so these transition states, these altered states, they can be there can be more than one altered structures. Also, these transition states are unstable. They are unstable. Okay, now if we talk about the word stability, stability is concerned with activation energy. Is concerned with activation energy. And what is activation energy? To initiate any chemical reaction, we need external supply of energy, which is known as activation energy. What is activation energy, beta? External supply of energy required to initiate a reaction, that is known as activation. Now see, we have a lot of chemical reactions. We have numerous chemical reactions. Writing it on the next page. Okay, we have numerous chemical reactions. To initiate. We have numerous chemical reactions in our body that require activation energy. And it is not possible to provide each reaction with large amount of activation energy. Basically, what enzyme will do, enzyme will lower the activation energy. Enzyme will lower the enzyme lowers the activation energy. Enzyme lowers the activation energy, and then we get this graph. This graph. Okay. So on x-axis, sorry, on y-axis we have potential energy, and on x-axis we have progress of reaction. Now what happens, say let's say we have substrate here. Okay, so if there is absence of enzyme, if there is absence of enzyme, the activation energy which is required by a chemical reaction will be this much. Then it will form the product. Okay, and when we have an enzyme, the substrate will have will have activation, this much activation energy. Only this much activation energy will be required by a chemical reaction form a substrate, so to form a product. Okay, so basically when enzyme was present, it lowered the activation energy. I hope you have clarity about this topic now. Okay class, now let's study about the factors affecting enzyme action. Let's study about the factors affecting enzyme action. Okay, beta. Factors affecting enzyme action. The first factor that we are going to study that affects enzyme action is temperature. What happens with the temperature? Enzymes work efficiently. Enzymes work efficiently in narrow range of temperature. Narrow range of temperature, which is known as optimum temperature. If 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 the temperature goes above this optimum temperature, the enzyme get denaturates. Okay, and if temperature is lower than optimum temperature, then enzyme gets inactivated. Okay class, and if I show you on this on the graph, okay, so here on x-axis we have enzymatic activity. This graph we have enzymatic activity, and on this we have temperature. Okay, so as the temperature increases, the graph goes up, then it goes down. Okay, so the enzymes work efficiently on this range of temperature, at this range of temperature. Okay, beyond this okay, lower than this optimum temperature, it gets inactivated, and above this temperature it gets denatured. Okay, Then if we talk about pH, works get enzymes get enzymes work efficiently in narrow range of pH. Okay, every enzyme have specific pH to work efficiently. Okay, and the okay, its graph is like the same as of temperature. Let's see its graph. We talk about the graph of pH. We have enzymatic activity. Then we have, okay, and the same graph will go here. Okay. Now the third factor is substrate concentration. Third factor is substrate concentration. Okay. As we increase the substrate, the enzymatic activity increases. As we the substrate, rate of reaction increases. And ultimately, the rate of reaction reaches its maximum velocity that is Vmax, maximum velocity that is Vmax, and does not rise after furthermore substrate. Why this is happening? That at some point when we are raising the substrate, when we are increasing, when we are adding the amount of substrate, okay, when we are increasing substrate concentration, the rate of reaction increases. Okay, but after some time it reaches Vmax, and then it won't rise furthermore. If even if we are adding more and more substrate, it will remain constant. Why it is happening? Suppose we have an enzyme here. Let's say we have an enzyme here. These are having two active sites. One is this active site and second is this active site. Okay. Now what happens if we add a substrate here? The rate of reaction increases. Okay. Furthermore, if I add a substrate here, the rate of reaction increases furthermore. Okay. But after this, if I want to add a third substrate, the reaction won't increase. Why it won't increase? Because they do not have a free active sites now. The active site that enzyme had, they are fully saturated now, and there is no more active site to accommodate this third substrate. Okay, so that is why after adding these substrate molecule, the enzyme the rate of reaction reaches Vmax, and after, if I want to add more substrate, and if I want to add more substrate and want that rate of reaction increases furthermore, it won't happen. Even after adding more and more substrate, the rate of reaction won't get increased because it has already reached its maximum velocity. Okay. The reason is active sites of enzymes are fully saturated. Okay, what is Michaelis constant? What is Michaelis constant? Plus, to reach the Vmax, we need amount of substrate, we need some specific concentration of substrate to reach the Vmax. Okay, so the amount of concentration which is required to reach the half of Vmax is known as Michaelis constant. Okay, so what is Michaelis constant? It is basically mathematical derivation. It is basically mathematical derivation which indicates the substrate concentration of chemical reaction catalyzed by an enzyme required to reach half of Vmax. That is known as Michaelis constant. And if I draw the graph here, let's say we have this. Okay, here on x-axis we have substrate concentration. Okay, and on this y-axis we have rate of reaction. Okay, here we have rate of reaction. Okay. Now, as here we have Vmax. Here above we have Vmax. Here we have Vmax. At this point we have Vmax. Here we reach Vmax. Okay, and this is on y-axis we have rate of reaction. Okay, now as we increase the substrate concentration, the rate of reaction increases. Okay, and up to a certain limit. Okay, after that it reaches the Tmax. Okay, and after that rate of reaction becomes constant. Okay, so to reach the Vmax we need a particular amount of substrate concentration. Okay, now to reach the half Vmax, here we have half Vmax. Let's say at this point we have half Vmax. Here at this point we have half Vmax. Half Vmax. Okay, here we have half Vmax. So now to reach the half Vmax, we require some amount of concentration. So the amount of concentration that is required to reach the half Vmax, that is known as Km or Michaelis constant. Michaelis constant is also known as Km. Okay, this Km is known as Michaelis constant. That's all for today's class video. Okay, So that's all for today's class. I hope I've made myself very much clear to you all with this enzyme topic. Okay, so we have discussed about basics of enzymes, and we have discussed about chemical reaction. Then we have studied about factors affecting enzymatic reaction. We have studied about activation. Furthermore, we have to discuss much more about enzyme, and we'll discuss it in the next lecture. I hope I've made myself very much clear with this lecture. Okay, so if you have any doubts regarding this lecture, please comment down. Okay, so we'll discuss the doubts in next lecture. Okay, I hope you have all understood this lecture now. I hope it was very much useful to all of you. Okay, bye everyone, take care.